Hello students, welcome to the second lecture of the chapter Molecular Basis of Inheritance. So in the last class, we talked about these following concepts, right? We talked about the structure of the polynucleotide chain or the nucleic acid, right? Uh, Means what all groups are there, what is a nucleotide, right? Then how two nucleotide form a phosphodiester bond to form dinucleotide and similarly polynucleotide. Then we also talked about the features of the DNA structure, that DNA is a double helix structure, and also what kind of nucleotide is there in DNA, what is the structure of the nucleotide present in the DNA, structure of the double helix itself, like what is the pitch, what is the distance between two base pairs. We also talked about base pairing rule and how the two strands are complementary to each other and anti-parallel in nature, right? Then we discussed what is the central dogma of molecular biology, which basically shows how genetic information within the cell flows from DNA to RNA and then to protein. And for this to happen, there are processes involved for each step. And lastly, we talked about how such a long strand of DNA, like in humans, the length of the entire DNA is around 2 meters, and how it is packed in such a way that it can be accommodated in nucleus, which is only 1 micron in size, right? So the packing happens in hierarchical manner, right? So we also discussed about that. Now, in this class, we'll be talking about how DNA was discovered as genetic material. Okay, so it's a quite a nice story, and I'll explain it in a story form, like how people did it. So intentionally, I'm going bit slow in this chapter because the concepts that you're studying here are a bit complex and you're studying for the first time. So I request you guys to take your time in understanding thoroughly each concept that is being taught. After watching the lectures, please make sure to read the books and make your own notes so that you learn everything properly. So let's start the story, like how DNA was discovered as a genetic material. So in the mid 90s century, okay, well, mid 90s, we talked that Miescher, he discovered nuclein, right? The nucleus, which we now know. And also Mendel gave the famous principles of inheritance, right? The principle, law of dominance, law of segregation, the law of independent assortment, which we talked about in the previous chapter, right? So in the mid 90s, all this work was done. Now, but they didn't tell us, or their work didn't indicate what is the biochemical nature of the genetic material. So this question was still yet to be answered. Okay, so it was left to the scientists after them to figure it out. So the work by Thomas Morgan, who worked with Drosophila, then Sutton and Boveri, who gave the chromosomal theory of inheritance. So work by these people actually narrowed down the search for the biochemical nature of the genetic material up to chromosomes. So they knew, so the scientists who worked after these people, they knew that we have to look for the genetic material within the nucleus, as people have found out that genetic material is present in the chromosome and chromosome is present in the nucleus. So the search was narrowed down to the nucleus. People knew in nucleus itself we will find genetic material. Now let's start with the first story. So this work was done by Frederick Griffith, okay, in 1928. So hello students, welcome to the second lecture of the chapter Molecular Basis of Inheritance. So let's recap what we have understood from the first class. We saw what is the structure of the polynucleotide chain or the nucleic acid, right? So it is made up of nucleotides and various nucleotides, they're bonded together by phosphodiester bond and form the polynucleotide. And also we saw what are the components present in a nucleotide. Then we know that there are two types of nucleic acid, RNA and DNA. And we focused on understanding what is the structure of the DNA in the previous class, right? We talked about the double helix model given by Watson and Crick, how the two strands of the DNA are antiparallel to each other and complementary, right? We have to apply base pairing rule, right? We talked about that, Chargaff's rule. Also, we talked about what is the pitch of the double helix structure, what is the distance between two base pairs, right? Then we talked about how the flow of genetic information happens within the cell. That is, we talked about the central dogma of molecular biology. We know that genetic information flow happens from the DNA, okay, then to RNA, and then to protein, okay. And in between, there are processes involved that make it happen, which we'll discuss soon in this chapter. And lastly, we discussed or we answered the question how such a long DNA strand, which is around 2 meters in length in humans, it is able to fit inside the nucleus of the cell, which is only 1 micron in size. So how such complex packing is possible? We talked about that, correct? We talked about the proteins that are involved in packing and what is the structural hierarchy that is present. So ultimately, we see, the packing of the DNA. Now, in this class, we will discuss uh, how genetic material or how did people prove that DNA is genetic material. Okay, DNA is genetic material. How did people prove? So there is a nice story that we will look at in this class, and it's actually a story. It's very easy to relate if think of it like a story. And intentionally, I am going slow for this particular chapter. I'm taking few topics in one class so that it is easier for you to go back and read your book and, make your own notes and, digest all the concepts taught smoothly. Okay, you shouldn't have any means ambiguity when we move on to the next topics, like subsequent topics. All right, should be able to recall each of the topics that are taught in the previous classes. So please make a note of that. So this story begins with the discovery of nuclein by Mouchandra, which I told you in the last class. Then discovery of the principles of inheritance given by Mendel in the mid-90s. So we know Mendel gave this. So, but these two discoveries didn't yet answer what is the biochemical nature of the genetic material. Okay, whether it's a protein or it's a DNA or RNA, like, or lipid, like what is the biochemical nature of the genetic material? It was not yet answered. Uh, then the work of Morgan, Thomas Hunt Morgan, who worked with Drosophila, right? We talked about linkage and recombination. So there also the work of Sutton Boveri, who gave the chromosomal theory of inheritance. So because of their work, the scientists who were going to answer this question, they knew to look for genetic material in the nucleus. Okay, because they knew the genetic material was present in the chromosomes, which is itself present in the nucleus. So the search for genetic material was narrowed down to the nucleus level. So they knew it is present in the nucleus itself. So their work helped in narrowing down the search. Now, how the search began? So first work, which is mentioned worthy, okay, is by Frederick Griffith. He conducted an experiment in 1928 using the bacteria Streptococcus pneumoniae. Now, this bacteria causes pneumonia. Okay, pneumonia is a disease. This bacteria is responsible for the disease pneumonia. Now, this bacteria has two strains that are found in nature. First is the S strain, second is the R strain. The speciality of S strain is that it has a smooth, shiny, it forms actually smooth, shiny colonies because it produces a mucus coating over the bacterial cell wall, or a polysaccharide coating. Mucus is basically a polysaccharide, and this coating over the bacterial cell wall gives a smooth gives or produces smooth shiny colonies. Okay, whereas our strain, they produces rough colonies. So when people looked at it, the colonies looked a bit rough, porous, where the colonies for the S strain was smooth. Our strain didn't produce this mucus coating over the cell wall, because of which they produce rough colonies. So because of the type of coating, where one had a mucus coating and one didn't have mucus coating, S strain was virulent. So what do you mean by virulent? Virulent means it can cause pneumonia. Okay. Whereas our strain is non-virulent, means it cannot cause the disease pneumonia. So he did two sets of experiment. All right. So he took one group of mice and injected them, like this is one group of mice. They injected them, like Griffith injected these mice with the strain Streptococcus pneumoniae bacteria. All right. And then he saw, because of the development of the pneumonia disease, these mice died. All right. These mice died. Whereas in the second group of mice, they were injected with R strain Streptococcus pneumoniae bacteria. And since we know R strain does not cause pneumonia, so these mice lived. Okay, it was simple enough to understand. One strain killed all the mice, S strain killed all the mice, whereas R strain didn't kill the mice. The mice lived. Now in another set of experiment, what he did, first he killed the S strain bacteria by heating. All right, by heating, the S strain bacteria were no longer alive, and he made again two groups of mice. Now, first group of mice, they were injected with this heat-killed S strain bacteria. All right, and since these S strain bacteria were no longer alive, they couldn't cause the pneumonia disease, so these mice lived. All right. Whereas in another group of mice, they were now injected with, listen carefully, they were now injected with the heat-killed S strain bacteria and the R strain bacteria. So a mixture of heat-killed S strain bacteria and R strain living R strain bacteria, they were living alive R strain bacteria. This mixture was made, and this mixture was injected in this group 2 mice. And he saw that these mice died. All right, they died. So why did they die? All right, uh, it is yet to be answered. So when Griffith did autopsy of these dead mice, he found living S strain bacteria. See the contradiction. So in group 2, the mice, they were injected with heat killed, means no longer alive S strain bacteria, and a few R strain bacteria, they were alive. All right. And this mixture killed the mice. And during autopsy, he saw that these mice, they had living S strain bacteria. Initially, he didn't give live S strain bacteria, but after death, he found live S strain bacteria. So what had happened here? This was the question. So he gave a very simple explanation, which is he said that the R strain bacteria, they had somehow transformed. They had changed their identity altogether. Okay. To S strain bacteria, because initially he gave live R strain bacteria and heat killed S strain bacteria. So, but in the dead mice, he found only live S strain bacteria. So he's saying this live S strain bacteria are nothing but the live R strain bacteria, which was given initially, but now these R-strain bacteria had transformed itself and became S-strain bacteria. So what was causing this transformation? All right. So he said that it is because of transferring or a transfer of a transforming principle, some transforming principle, something, okay? Something is getting transferred from the heat-killed S-strain bacteria to R-strain bacteria. And because of this transfer from heat-killed S-strain bacteria to R-strain bacteria, the R-strain bacteria had lost their original identity and became live S-strain bacteria, which were virulent in nature and could cause pneumonia. And they formed colonies. They formed shiny, smooth colonies because of the production of, or because of the synthesis of mucus coat. So he said this transforming principle that is getting transferred, all right, from the dead S-strain bacteria to live R-strain bacteria is the genetic material. All right, genetic material, because the transfer of genetic material, the R-strain bacteria has lost its original identity and became S-strain bacteria. But his experiment still didn't confirm what is the biochemical nature of genetic material. Means what is the biochemical nature of this transforming material, or of this genetic material that is getting transferred from dead S-strain bacteria to live R-strain bacteria. So this was Griffith's experiment, where he showed because of the transfer of genetic material from dead bacteria to a live bacteria, the live bacteria changed its identity, okay, and showed a property which was not its original property. All right. This experiment is also called transformation experiment, which people in the field of biotechnology uses this experiment to change the identity of a bacteria. So after Griffith's experiment, there was assumption by the biologist at that time that genetic material is in fact a protein. All right. But this was proven wrong 2 times actually. First time, biologists didn't believe that it is true. All right. Second experiment, they couldn't argue anything and they had to accept that protein is not the genetic material. So what were the 2 experiments? The first experiment was done by Avery, McLeod, and McCarthy in 1933 to 1944. So what they did the extension of Griffith's experiment. Okay, so they tried to analyze genetic material of the transforming principle which Griffith proposed. So they took heat-killed S-strain Streptococcus pneumoniae bacteria, all right, and from the dead bacteria, they purified all the biochemicals which are present: DNA, RNA, and protein. These 3 biochemicals were purified, all right, and they were tested one by one, uh, and proposed which could be the transforming principle, or which could be genetic material which is causing the transformation of live R strain to live S strain. All right. So from the heat killed S strain bacteria here, first they took its RNA and injected into the R strain bacteria and that the saw that the R strain didn't transform. It didn't transform into S strain. And then next they took its protein only. They took the purified protein from the heat-killed S-strain bacteria, all right, and then again injected into live R-strain bacteria, the second one. And again, they saw there was no transformation, means the R-strain bacteria didn't transform into S-strain bacteria. Now, in the third case, they took purified DNA from the heat-killed S-strain bacteria and injected into R-strain bacteria, live R-strain bacteria. Now, in the third case, they did saw transformation. This worked. These two didn't work. So now they concluded that DNA is in fact the genetic material or the transforming principle which caused the transformation or the change of the identity of the R strain bacteria to S strain bacteria. Now, they proved the same thing, okay, in another way, okay. So what is the another way? They now took a mixture of DNA, RNA, and protein from the heat-killed Estrian bacterium. They took a mixture. Now, in this mixture, first, they added RNAs. To this mixture, they added RNAs. Now, what is RNAs? RNAs are proteins, hydrolytic proteins or hydrolytic enzymes that digest RNA. So if you add RNAs into um, into a RNA mixture, the RNA will be degraded altogether. Okay. After some time, you won't find any RNA into that solution. All right. So in the first set, when they added RNAs to this particular mixture, they saw, as I said, you would expect RNA to be deleted, means it will be degraded altogether. So there will be only DNA and protein present. So then the first set, and when this mixture, RNA, DNA, and protein plus RNAs this mixture was injected into the into the R strain bacteria, they saw that the R strain bacteria did transform to S strain bacteria, which means transformation of one strain of bacteria to another was not affected in presence of RNAs. All right. So they concluded from his that RNA cannot be the genetic material. All right. Because presence of DNA or protein, either of these is in fact genetic material which is causing this transformation. Now in the second set of experiment, instead of adding RNAs, they added proteases. Now these are again hydrolytic enzymes which digest proteins. So in presence of proteases, protein will be digested. So in this particular mixture, we will have only DNA and RNA then. Now when this particular mixture will be injected into R strain bacteria, live R strain bacteria, they saw the transformation did happen from R strain to S strain bacteria. So now we can say that protein is also not the genetic material. Now lastly, they made another mixture. Now in this case, they added DNAs, which is an hydrolytic enzyme that digests DNA. Now in third case, DNA will be digested. Only RNA and protein will be present in the mixture. Now when this mixture was injected in R strain bacteria, they did not transform to S strain bacteria. Transformation did not happen. Okay. Now this third set conclusively proved that DNA is in fact the genetic material which caused the transformation, as either of the above two sets, in either of the, in set 2 and set 3, transformation did happen, right? Whereas in set 3, transformation didn't happen because DNA was not present. In set 1 and set 2, DNA was present. Although protein or RNA was degraded one by one, but DNA was present. So they said that DNA is in fact a genetic material. So they proved the same thing, okay, through two sets of experiments which are similar to one another. But still biologists were not convinced, all right? They were not convinced with the study, as I showed, that DNA is genetic material. To many of the biologists, they still thought that protein could be the genetic material, okay? And they didn't believe the people who are saying DNA is genetic material. So there was a debate going on. What could be genetic material, whether it is protein or whether it is DNA. All right. So this debate was ultimately resolved by Hershey and Chase, all right, in 1952. And they proved without any doubt, unequivocally means without any doubt, that DNA is in fact genetic material. How did they do? They did it through an experiment, obviously. So what was their experiment? So Hershey and Chase, they were working at that time with bacteriophages. Now, bacteriophages are viruses, are a type of viruses that people still use. And these viruses, specialty of these viruses is that they can infect bacteria. They are not coronavirus, they are bacteriophages, and they can only infect bacteria. They don't infect us. Okay, they don't infect the mammalian cells. They only infect the bacteria. So how they infect? The mechanism is quite easy actually. So a bacteriophage first attaches to the bacterial cell wall. All right. It goes ahead, like if this is the bacteria. All right. Let's say this is the bacteria and something like this is the phage. It has legs and it will, using those legs, it will sit or attach strongly to the bacterial cell wall. Then what it does, it transfers, here genetic material, something is present. The genetic material is transferred into the bacteria. And this genetic material goes ahead and integrates with the bacterial genetic material. So bacterial genetic material treats this viral genetic material as its own and allows integration of the viral genetic material, because of which we see that inside the bacteria, or bacteria becomes a house or a factory for new viral particles to be formed. It becomes a factory where new viruses can be formed continuously from the genetic material of the virus that has been integrated with the bacterial genetic material. So that is how the infection, or that is how bacteriophages infect the bacteria. Right? The mechanism is quite easy. First, it attaches on the bacterial cell wall. Then it transfers the genetic material into the bacteria. The genetic material of the virus becomes integrated with the genetic material of the bacteria. All right. And then the bacteria cell becomes a factory for the synthesis of new viruses or new bacteriophages. So in this way, it is using bacteria as a host for, making new and new viruses. So in this figure, this is taken from your book. This figure explains what was Hershey and Chase's experiment. So yeah, they took bacteriophages, all right. One group of bacteriophage was grown in a media containing radioactive S35, containing radioactive, it is radioactive in nature. So one group of bacteriophage was grown in a media containing radioactive sulfur, all right. And another group of bacteriophage was grown in a media containing radioactive phosphorus. Now this is an isotope of phosphorus and this is an isotope of sulfur, which is known to have radioactive properties. Now, if you recall the structure of the protein and the DNA, we know protein will become integrated into the backbone of DNA, right? The backbone of DNA is made of sugar and phosphorus or phosphate groups. So this radioactive phosphorus will become part of the DNA, making DNA of the virus radioactive. Whereas sulfur, we know, is present in amino acids like cysteine, right? So it will then, and when these amino acids will form peptide bonds, will form the polypeptide of the protein. So sulfur will then become part of the protein of the virus, and this protein will be radioactive in nature. Now, virus is a very simple organism, all right? The virus has a coat, okay, shield, and this coat is made up of protein. And within this coat is the genetic material, okay? Now, um, and the genetic material, they are assuming, let's say it is DNA, all right? We have not yet proven what is the genetic material, but now we know it is DNA. So coming from, modern view, I'm trying to explain what is the structure of the virus, all right? So now we know DNA is genetic material and it is present inside the protein coat of the virus. So that is how the virus looks like, the bacteriophage looks like. Now please divert your attention to this particular figure. So these are two groups of bacteriophages, right? Group 1 and group 2. Now group 1, in the group 1 bacteriophage, the protein is radioactive in nature, means the coating, all right? The red color is coating, means this diamond shape, this shape, this diamond shape, and then the legs, they're all made up of protein, and uh, that is radioactive, okay, because of the integration of the radioactive sulfur. But in the second group, the genetic material is radioactive. Um, that is DNA is radioactive, all right, because of the integration of the radioactive phosphorus. So they labeled both DNA and protein separately in two groups of bacteriophages. Okay, they labeled radioactive using radioactive isotopes. Now, when these two groups of bacteriophages were one by one allowed to infect E. coli or Escherichia coli, which is a bacteria. Okay, E. coli. Now, these bacteriophage were allowed to infect two sets of E. coli. Now, in the first set, where the bacteriophage had radioactive protein, so it attached to the green color bacterial cell wall, all right? And in the second group also, bacteriophage will now attach to the green color cell wall. This is the infection step. Then what they did, after we know mechanism how the infection happens, we know that after attachment, the bacteriophage will transfer its genetic material into the bacteria. So after it will do that, the bacteriophage viral coat, which is protein, okay, it will be completely empty. So they did blending, okay, to remove or detach the empty protein or viral coat from the bacterial cell wall. So you can see the structures are now being removed. Okay, previously in the previous step it was attached to the cell wall for and facilitated the transfer of genetic material from the virus into the bacterial cell. The second step, after the transfer of genetic material has happened, the blending process allowed detachment of the viral coating. Okay, now they had a solution which contained a mixture of viral protein coats and the bacterial cell, and this bacterial cell has the genetic material of the virus. Now they did centrifugation. Okay, they did centrifugation. Basically, they used a machine where this mixture of viral coat and the bacteria was spinned, was rotated at a very high speed, or at a very high revolution per minute, or RPM. So that allowed separation of two layers, formation of two layers basically. One is called the supernatant, which is lighter, okay, and it is towards the this is the tube, say. So this is the lighter portion. This is called supernatant, represented with S, and the bottom one, which is heavier, it will be called pellet. So when the centrifuged, they got a lighter supernatant that contained the viral protein, okay? And in the pellet, they got the cells which are heavier in nature, bacterial cells. And then they examined the viral protein and the bacterial cells individually. So what they found? They found that in the supernatant of the first group, supernatant of the first group, uh, the bacterial, uh, it's a viral coat will be there, and that viral coat would be radioactive in nature, right? Because in the first group, the bacteriophage were grown in the presence of radioactive sulfur, and the sulfur got integrated into the protein. So in the supernatant, in the first group, they found radioactive protein, radioactivity in the supernatant, where in the pellet, where there was the bacterial cell, there was no radioactive detected. All right, means radioactive isotope S35 was not present inside the bacterial cell, so it was not radioactive in nature. Where the supernatant had the viral protein coating, all right, and it was made up of radioactive sulfur, so radioactivity was detected in the supernatant. On the second group, where bacteriophages were grown in the presence of radioactive phosphor P32, the supernatant did not have radioactivity, right? Whereas the pellet had radioactivity. So again, I'm repeating, the supernatant will have the viral protein coat, and phosphorus will not be present in the protein, it will be present in the DNA, right? So whereas the pellet will have the bacterial cell. Now we know during infection, the bacteriophage had transferred its genetic material to the bacterial cell. So the bacterial cell now houses the radioactive viral genetic material, that is radioactive viral DNA. So that is why the supernatant bacterial cell, they were radioactive in nature. So this proves conclusively that it is in fact the DNA is what is the genetic material, okay, and because it is the DNA that is getting transferred from bacteriophage to from bacteriophage to bacterial cells. It is not the protein which is getting transferred from the bacteriophage to bacterial cells, E. coli cells. All right. And as I previously told, mechanism of bacteriophage is such that during infection, it transfers its genetic material, right, from and transfers genetic material inside the bacterial cell. And, and the result of this experiment proved genetic material is in fact the DNA, right? And you saw that in group 2, the pellet, the bacterial cells were radioactive in nature because they had the radioactive viral DNA in them, right? So this was Hershey and Chase's experiment, and this type of experiment is still done by biotechnologists, and this experiment is called transduction, where a bacteriophage, there are various strains of bacteriophage as well, which is used for transferring particular DNA segment, okay, into the bacterial cell, right, or into a mammalian cells as well. So this is the transaction experiment which Hershey and Chase did to prove that DNA is the genetic material. And after this experiment, no other biologist argued against it, and everyone agreed that DNA is genetic material, not the protein. So the initial debate was over. Now let's talk about what should be the property of genetic material, or what criteria uh, like biomolecule, that is DNA or RNA, okay, a nucleic acid should have, which makes it a genetic material. If that nucleic acid, DNA or RNA, has any four of these criteria, then we can say yes, it can act as a genetic material. All right. So people observed that DNA is genetic material of majority of the living organism. I told it yesterday in my previous class as well. Whereas RNA is genetic material in some viruses like various bacteriophages, etc. So why is that? All right, that is also a thing that can be answered from each of these four criteria. So what is the first criteria for nucleic acid, DNA or RNA, to be a genetic material? First criteria is that it should be able to generate its replica. All right, means DNA should make copies of itself. Like from DNA we should get DNA. Similarly, RNA should make copies of itself. All right, and we know that there are processes, okay, biochemical processes through which both DNA and RNA can direct its duplication. All right. So first criteria that it should be able to generate its replica was fulfilled is like can be fulfilled by both RNA and as well as DNA. Second criteria is it should be stable in nature. What do I mean by stable? I mean the chemical structure as well as the physical structure. So when we look at this particular point, we can now make a quick distinction why DNA should be preferred over RNA. So if you recall from my previous class, the first class, that DNA is a double helical structure. All right. So DNA is a double helix. So two DNA strands form the double helix. So this gives a physical stability, a structural stability. It is quite easy to cut one strand of DNA, but if two strands are present, the job of cutting becomes a bit difficult. So it is not easy to cut a double stranded DNA strand. All right, it makes the job of degrading double stranded DNA difficult, whereas we can easily degrade a single stranded DNA. Whereas RNA is always single stranded. It exists in nature as single stranded. We can easily cut RNA strand. So structurally, DNA is more superior than RNA. Now, what about chemical structure? So we know RNA contains ribose sugar and DNA contains deoxyribose sugar, right? And if you recall, in the ribose sugar, there was OH group in the 2' end, all right? So because of this particular feature, RNA is more structurally, chemically unstable. It is very reactive in nature, all right? Whereas DNA is not that reactive. So if people have seen that inside the cell, uh, the RNA can act as a catalyst, all right, and catalyzes various reactions. Uh, so and your catalysts are itself have a very reactive property. So presence of this 2' OH is giving that catalytic property to various RNAs, whereas DNA cannot act as a catalyst, it comparably is chemically more stable. Also, RNA contains uracil, right, as a base, whereas DNA instead of uracil there is thymine. So this distinction between the type of base is also responsible for the chemical stability of more chemical stability of DNA than RNA. So uracil is giving RNA less stability property, all right? It makes it more susceptible to degradation, whereas thymine in DNA makes DNA structure much more resistant to degradation. So also RNA is a very, very reactive molecule. It can be degraded very easily. So people who are working with RNA molecules in labs, they have to be very, very careful, because after all the work they do for isolating the RNA, if they become a bit careless, they can lose the sample, RNA sample altogether. All right. Whereas comparatively, it is easy to work with DNA molecules. So from through this argument, one can conclude that DNA is more stable both structurally as well as chemically than RNA. Next criteria for either DNA or RNA to be genetic material is that it should provide the scope for slow changes. Also, the slow changes are nothing but mutations that are required for evolution. So in the previous chapter, we talked about what are mutations. The mutations are the permanent change, all right, or permanent alteration in the basis of the DNA sequence. So how mutation happen, etc., we will study more in detail in this chapter as well. Also, how mutation are the seed for the evolution, which will be discussed in the next chapter, evolution. So what I'm trying to say that the genetic material should allow some changes, okay, but the changes should not be too rapid. It should be slow in nature. So as I told you, RNA is a structurally or chemic and as well as chemically much more unstable in nature than DNA. So organisms like viruses who have RNA as the genetic material, they mutate quite fast. So coronavirus or COVID-19 is known to have RNA as genetic material. All right. So people are fighting one strain of coronavirus, but in few months or few days, another strain of coronavirus emerges. Why? Because RNA mutates quite fast. It is structurally unstable. It allows various environmental factors to mutate it mutates its sequence to change its sequence, because of which a new strain of coronavirus has emerged. So, whereas DNA, we have all the humans all, and various multicellular organisms, they have DNA as the genetic material. Now, DNA do allow change in the sequence, but the change it's very slow, right? Comparatively very slow. So that is why for a new species of any organism to arrive or to develop in nature, it takes thousands of years. All right, we can in few months we cannot see a new species of human, right? So this to this point we can see that both RNA and DNA allows mutation, but in RNA the mutation is very fast, because of which new strains of viruses, etc., emerged very quickly in comparison to those organisms having DNA as genetic material, and there the mutation happens very slowly, like one in million bases. Okay, one in million bases gets changed or gets mutated. All right, and then it takes thousands of years for that mutation or a group of mutation to give rise to a new species of human altogether. So that is what we'll talk about in the next chapter, evolution, like how new species are formed, what is natural selection, etc. So this point is clear, all right. Then the fourth point is, or the fourth criteria for DNA or RNA to be genetic material is uh, it should be able to express itself in the form of Mendelian characters. Means we saw in the previous chapter that Mendel proposed that there are factors, right? In his terms, factors, which we now genes, they get passed on from one generation to next generation, because of which the progeny shows the parental characters, all right? So this kind of behavior should be displayed by DNA as well as RNA. So now we know that both DNA and RNA can do that, all right, but it is much more direct for DNA to do that, all right. RNA has to use a enzyme or a biochemical reaction to convert it to make copies of DNA. Means RNA has to make copies of DNA, and this DNA can be inherited to the next generation. So nature has favored the inheritance of DNA, all right, from one generation to the next generation, and it didn't favor much about inheritance of RNA to DNA. Whereas RNA is a much more useful nucleic acid when it comes to expression, all right, expression of characters, expression of Mendelian characters like tall, dwarf, etc. So it is RNA actually that directs the formation of proteins, and we know proteins are involved in various biochemical reactions, all right? They are the enzymes, they are the hormones, they are the antibodies, all right? They make up various structural components in cell, extracellular matrix of the tissue, etc. So protein is a very important part of our body. So it is the RNA that direct the synthesis of protein. If you recall the central dogma of molecular biology, we talked about from DNA RNA can be made, and from RNA, protein can be made. So we are here. So in this statement, the statement that RNA can direct protein synthesis is actually pointing to this particular section of the central dogma of molecular biology. So it is the RNA that can direct protein synthesis, and DNA is actually dependent on RNA for this. Okay, it's actually dependent on RNA for protein synthesis. DNA cannot directly made protein. This is not allowed by nature. There is no mechanism that allows direct synthesis of protein from DNA. It has to go via RNA. Okay. So RNA is equally important. All right. So all these criteria 1, 2, 3, 4, we saw a comparison, right? Which comparison between RNA and DNA, and these four criteria conclude that DNA being more stable, all right, being able to generate its replica, as well as it allows slow mutations, and it can be inherited from one to next generation. So DNA is a much more suited material for and is a much more suited genetic material actually than RNA. All right, but in nature there are few viruses, they do have RNA as the genetic material, whereas or the higher multicellular organisms like us, they all have DNA as genetic material. So because of this stable structure, DNA is much more preferred for storage of genetic information, which now can be transferred from one generation to the next generation, and flourishing. And we know how genetic information get transferred, right? Meiosis process happens, etc. And it is a DNA which is present in the chromosome itself, in the chromosomes that people have also discovered. So that's the end of today's discussion. All right. So this lecture was kind of a story which told you about how DNA was conclusively proven by scientists as the genetic material universally. All right. Um, Not one, but 3, 4 experiments we talked about. Right. One was the transforming principle. Right. There's something getting transferred from the heat killed R strain bacteria to S strain bacteria. And what is getting transferred? It is the DNA getting transferred, which later on proved by Avery, McLeod, and McCarthy. All right. Then transduction experiments was done by Hershey and Chase. They also they actually resolved the debate between DNA and protein, like which could be the genetic material. And their experiment showed that it is the DNA which is getting transferred from bacteriophage to bacterial cells, because of which the bacterial cells were radioactive in nature, where in the group where the protein was radioactive in nature, there the bacterial cells didn't show any radioactivity. So these were few experiments that proved DNA is genetic material. Now the argument is there, which is a more suited genetic material, okay, whether a DNA or RNA. So we discussed about these criterias, all right, for RNA or DNA to be a genetic material. So all these 4 criterias can be beautifully applied to DNA. All right. RNA meets few of these points, obviously, but it also have its own limitations. So because of which all higher organisms, multicellular higher organisms, they have DNA genetic material, whereas primitive organisms like viruses, etc., they have RNA. So in the beginning of the time, all right, when life started flourishing, people said that RNA is the first genetic material which was present in the primitive organisms, just like the cells. And these RNA actually preferred, all right, by the nature, which then from RNA directly proteins were made and the structure of the cell was developed, all right. But later on, nature identified the limitation of the RNA structure, right? It is much more reactive. It is much more stable. So chemical modifications happened in the structure of the RNA, and then from RNA, DNA was formed. So that is how genetic material evolved. All right. Now all the higher organisms, multicellular higher organisms, they have DNA as the genetic material. So it is a very beautiful story of discovery of genetic material. In the next class, we will talk more about the DNA only, how it gets replicated, all right, how RNA is formed from DNA through transcription, then from how protein is formed from RNA through translation. So these all topics will be covered one by one in the subsequent class. Thank you for joining this class, and if you have any queries, you guys can always reach out. Thank you.